Thank you to Quanterix for inviting me to speak with you today. My talk is a little more technical, I think, than most of the other presentations, although the presentations today have been wonderful, just something a little bit different. So I'm going to speak to you today about the development of a sensitive homebrew SIMOA assay to detect LRRK2, which is leucine-rich repeat kinase 2. So first I'll give you a little background on LRRK2 and Parkinson's disease, talk about our assay requirements. We tested mesoscale discovery MSD platform versus SIMOA. And then I'd like to tell you about a unique collaboration that we have between Quanterix and the Michael J. Fox Foundation and Pfizer. We were recently awarded a grant, which we're very excited about. Then I'll get into some of the details about our homebrew assay development. Homebrew assay development can be quite challenging compared to using predefined kits from Quanterix, and specifically talk about assay, reagent, and instrument challenges. And then I'll go and show the utility of the assay for some cell lysates, as well as talk about measuring LRRK2 and cerebral spinal fluid. So Parkinson's disease is a motor system disorder, which is a result of the loss of dopamine-producing cells in the brain. The symptoms are, you get some trembling in the hands and the legs and the arms and the face, very stiffness in the limbs and the trunk of the body, very slow gait and instability in general. The age of onset is usually greater than 60 years old, although you can have earlier onset. There's sporadic Parkinson's or idiopathic Parkinson's versus familial Parkinson's. And the diagnostics, there are no known blood tests to diagnose Parkinson's as of yet. Right now, you go to your family practitioner, your general practitioner, they do some tests, they send you to a neurologist, and basically they do some brain scans to actually rule out any other types of disease that might have similar symptoms, such as a stroke, for example. So this is the LRRK2 protein. It is a very large, complex, multi-domain protein. It has a tricatalytic domain. It is a kinase, so this is the kinase region. There's a GTPase region as well. Some of these other portions of the protein are thought to be involved in protein-protein interactions. But pathogenic mutations are thought to trigger the pathogenic events that lead to the disease. And of particular interest is this G2019S mutation, which can cause an increase in kinase activity for LRRK2. So it's an attractive target to inhibit the kinase activity. These phosphorylation sites, S935 and S1292, are of particular interest. LRRK2 mutation carriers develop the disease that is very clinically similar to the sporadic Parkinson's. You'll see a lot of similarities in the symptoms. However, some differences are early onset of the disease, as well as more frequent gait disturbances and muscle wasting as well. So we were asked by our research unit to develop assays, sensitive assays, to quantitate LRRK2 in various matrices and in various species. The current assay format that they have is a Western blot, which is not very quantitative, it's not very sensitive, it's a cumbersome assay method, it has low throughput, it's not something that really you can go into the clinic with. So they asked us to develop a total LRRK2 assay and a S935 phosphospecific assay. And we're very lucky to work with the Michael J. Fox Foundation, which is an organization that's dedicated to treating and curing Parkinson's disease. And they work with academia and industry and patients to come together, and they actually bring together consortiums where, in this case, they've made lots of different assay tools to study LRRK2 for Parkinson's. There's assay reagents, there's animal models, there's in vitro potency assays, things like that. So we're very lucky that in about 2008, 2009 timeframe, there was already a really nice panel of anti-LRRK2 monoclonal antibodies that were available. And this information was widely available to the research community. So there's publications on these antibodies, and they've been very, very well characterized to be used in multiple applications. So not just in assays, but also for use in immunohistochemistry, immunoprecipitation, Western blots, things like that. So you need reagents as building blocks, right, to build your assay. And also you need a ligand binding assay platform. And so in our lab, we have multiple platforms to choose from. We have gyro labs, we have mesoscale discovery, We have SIMOA as well, and also Singulex Arena. So SIMOA and Singulex Arena are very ultrasensitive platforms. So we went about to actually build the assays and what platforms were we going to put this on. So we first started with MSD. And the advantages to MSD is you get a very broad dynamic range. It's pretty easy to use. It's just a plate-based assay platform. It has good precision, and you can actually get pretty good sensitivity on MSD, at least down in the low nanogram to picogram per mil range. Some of the negatives would be in some of the matrices that we wanted to measure LRRK2, such as CSF and urine, we were expecting very low picogram per mil to sub-picogram per mil levels. So MSD may not be sensitive enough, and it also is not an automated system. The Quanterix SIMOA offers a lot of the same attributes that MSD does. For example, it does have a broad dynamic range, and for us this was really important because measuring LRRK2 in PBMCs, you get in high nanogram per mil levels, but going down to CSF and urine, you would be down in the low picogram per mil range. So SIMOA, it has ultrasensitive capabilities that MSD does not. The disadvantages is that for us, this is a new technology, so we're pretty early adopters of this platform. We purchased 4 instruments for different Pfizer sites back in late 2014, which were implemented in early 2015. So we're relative newbies on this technology. So the ease of use, particularly for homebrew assays, it can be a steep learning curve depending on the assay reagents that you have. So we're not using kits in this case. And the cost of the consumables are just a little more expensive than using MSD as well. And this is just, again, another cartoon of the LRRK2 protein, and in knowing and characterizing all of these reagents, and this has all been published, we know what epitopes these different antibodies bind to. So this is a comparison. I won't show you all of the pair testing that we did, but just in looking at the best antibody pairs on the 2 platforms, you can see that this is the blue line and the red line are the MSD assays for the phosphospecific and the total assay, and then the green and the purple lines are the SIMOA assay standard curves. And you can see that the sensitivity range for SIMOA was 10 to 20 picograms per mil compared to 100 to 200 picograms per mil for the MSD platform. So in general, although the MSD afforded much better sensitivity than a Western blot, the SIMOA assay gave 10-fold improvement even over the MSD assay. And it also still provided that broad dynamic range that we needed to measure in the multiple matrices. Okay, so now I'd just like to tell you about the grant that was recently awarded jointly to Pfizer and Quanterix by the Michael J. Fox Foundation. And the title was The Development of LRRK2 Clinical Assays on the Quanterix SIMOA platform. This unique grant allowed us to fund a Quanterix application scientist to actually work in my lab at Pfizer to develop these LRRK2 assays. So this is a little different approach than what Danielle used in using the actual accelerator lab at Quanterix. We felt that we preferred to have a Quanterix scientist work directly with us so that we could move along the technology quicker and get the instruments really up and running and really learn how to do homebrew assay development. So we have Carlos Herman Cabrera, who's here in the audience, and he's working in our lab, and he's doing a great job on these assay development efforts. So this provides us with sensitive assay tools to support the LRRK2 inhibitor program at Pfizer. And this is a win for Parkinson's research, Pfizer, and the Michael J. Fox Foundation. We will, as part of the grant, publish these assay methods so that they will be available to the entire research community. And again, this helps us move the SIMOA technology forward at Pfizer for homebrew assay development. So one of the things I wanted to make you aware of or touch base on is some of the reagent challenges that we faced. I found this not just working with SIMOA, but also with the Singulex Arena. Our detector antibody, we obtained 3 different lots from a commercial vendor. And the 3 different lots, lots 1, 2, and 3, you can see that the background differences are, and this is a log scale on the y-axis, are 10-fold for one lot and then 100-fold difference for the other lot. And you can see this has a huge impact for sensitivity. And I've seen this time and time again. It's just, you have to be very careful about the reagent quality when you're working with ultrasensitive assays. So our reagents team did attempt to try to find a smoking gun and define what was the contaminant or if there were particular aggregates in these reagent lots. And we used SEC, SDS-PAGE, and IEF testing. And the only thing that we could really kind of really pick apart was, this is on an IEF gel looking at unbiotinylated and biotinylated reagents. Now these were biotinylated the exact same way using the Quanterix biotinylation procedure. And we found that this particular sample had a higher biotinylation ratio than the others, and this was lot 2, but lot 3 did not seem to have a higher biotinylation ratio. So again, not a smoking gun. We were able to titer this reagent down so that we could get it somewhat similar to the first lot, but there have been significant stability issues with this particular antibody, and so you just have to be careful to test lot-to-lot variability if you're purchasing these antibodies commercially. And I think just overall that the technologies that we use to try to determine what these contaminants might be that are causing these high backgrounds in the assay just might not even be sensitive enough to pick those up. So I think that these contaminants or aggregates are highly magnified on the ultrasensitive platforms. And then I just want to touch base very quickly on the SIMOA 2.0 upgrade. For those of you in the audience that have SIMOA instruments, I'm sure you got your upgrade late last year, just like we did. And I'm actually thrilled that Quanterix provided this upgrade in the non-pelleting assay format. So we have seen a lot of improvements by this. And in particular, we were really pleased to have the reduced sample and reagent volume requirements. For us, this was really important since I lead a discovery bioanalytical lab, which uses a lot of rodent samples, so we really don't have a lot of sample to play with. And then we also have seen an overall improvement in precision through the upgrade. Although we do still have some issues, I am learning, I've been doing assay development for a long time on a lot of different platforms, and I am learning, especially from Carlos working in our lab, that the default assay buffer conditions that you typically use for some of the plate-based technologies, or even some of the other bead-based technologies, can be very different when you use the Quanterix platform. So, for example, things like surfactant and different buffers, the surfactant could lead to a lot of bubbles on the image, and that could lead to a lot of these NAN results, which are not a number. It's an acronym for not a number. So the upgrade has helped with this. And the other thing I wanted to share with you is that when we do design of experiments to optimize our assay conditions to get the most sensitive assays possible, one of the critical factors that comes out is typically lower bead concentration is important for lower background and better sensitivity. However, then you reach that sort of critical bead fill threshold, which can lead to this too few beads exception. And so one of the things we've implemented is to start using the Quanterix helper beads, which the people in my group actually call them buddy beads. And they've really helped with improving precision because we like to keep our bead concentration kind of on the low side. We do still need to run triplicates, but I really like the NAN. I'm learning to embrace the NAN concept. I think it has really good diagnostic value in helping you figure out what's going on, and it gives you something to go back to Quanterix to. However, at the end of the day, you don't have a result. So I appreciate the fact that Quanterix is working on this and provides the customers with these really nice troubleshooting flow charts and videos. And I still think that customers, at least I would love to have even more training as an advanced user on how to use these NAN exceptions and try to diagnose what's going on with the instrument. So now I'd just like to go into some of the data that we've generated and the utility of the assay. So this is looking at PBMC lysates. These are human lysates plus or minus drug treatment. And this is a vehicle sample, and these are, so this is phosphospecific LARC2 S935 LARC2 measures versus total, and you can see with drug treatment in a dose-dependent manner you get a reduction in phosphorylated LARC2 at the S935 site. However, in the total assay, the concentration of LARC2 is relatively unchanged. And then we also looked at LARC2 measures, phosphospecific and total, in non-transgenic and G2019S transgenic mouse brain lysates. And the non-transgenic levels are pretty low. And then for the phosphospecific, this is a vehicle and this is drug treated, so the S935 levels are reduced, and the total levels are not reduced to the same amount. It is reduced a bit, although here we're not normalized by protein concentration, which is something that we typically do. And then looking in the preclinical space, we also wanted to make sure, or we hoped that the reagents would cross-react with dog LRRK2, so we could support some dog studies. And they do. The black lines the black bars are total LRRK2 measures, and the gray are phospho LRRK2 measures. This is pre-dose versus post-dose. You can see the total levels are the same, but you see a reduction in the phospho LRRK2 levels. And again, this is pre-dose versus post-dose. And then you see the increase in phosphorylated LRRK2 as the drug is washed out. And then I'd like to touch on optimization of sample collection procedure for human PBMCs. I know many in the audience are biomarker folks, and we know that collection sites may have limitations in their basically processing limitations. They may not have the resources to process things the way you want them to. They may not have the equipment across all of the different sites. And so it's important to show that the different procedures that you're trying out, that the integrity of the sample is maintained, and in particular, the phosphorylated state of the protein in this case, because we're looking at phosphorylated levels at a particular site versus total. Thank you. Yep, almost done, thanks. So in this case, we looked at several different collection procedures and at different temperatures and different collection tubes. So this is using EDTA and phycol separation, and this is a cell preparation tube at just immediately processed, and you can see phospho versus total levels. When you put everything at room temperature for a time, you get dramatically reduced phospho-LRRK2 and also total LRRK2. So room temperature was not a good idea. 4 degrees was largely okay. One of the other things that we looked at too, and exosomes has come up several times in several different presentations. So we did measure LRRK2 in an exosome urine pellet. And they also wanted to have us try frozen whole blood, which we told them wouldn't work, but we tried it anyway. We just had a lot of debris and really no result. But if I show an inset of the urine exosome sample, we are able to detect LRRK2 in the urine exosomes in about the 100 picogram per mil range. Okay. And I did want to touch base very quickly on using cerebral spinal fluid. My take-home message here is shop around. The cost can vary from vendor to vendor hugely, from $30 per mil to $750. One of the things that we looked at was blood contamination, using a hemoglobin measurement as a surrogate for blood contamination. PBMC levels have very high LRRK2. If blood gets into your CSF sample, that could give you arbitrary high LRRK2 levels. And so this is just showing the expensive vendor at $750 per mil, had pretty high hemoglobin levels over the acceptance limit, 200 to 400 nanograms per mil, which is Michael J. Fox's recommended limit. And then this second vendor, which was only $100 per mil, was largely within the acceptable limit, and just 2 out of the 5 samples were right on the edge of acceptable. Then when you look at the hemoglobin levels measured in the individual CSF samples versus the total and phospho-LRRK2 concentrations, you'll see that largely we got below limit of quantitation values for CSF. We did get some positives, and we're trying right now to confirm that these are true positives. And then we also saw that the hemoglobin levels did not correlate with the LRRK2 levels, which didn't surprise us too much, because when you process the CSF, you spin out the cells, you should remove the PBMCs, but if there's some lysis going on, that's kind of what we were worried about. And then just in summary, again, we have this unique partnership between Michael J. Fox Foundation, Quanterix, and Pfizer, which is working very well. We've developed SIMOA fit-for-purpose assays for total and S935 phosphorylated LRRK2 with a 10-fold improvement over the MSD assay. We were able to demonstrate phospho-LRRK2 level suppression in isolated PBMCs and mouse brain astrocytes after treatment with drug inhibitor. And this provided confidence to our team that we can use this platform and that it will have the required sensitivity that we need for CSF. We will publish these assay methods in the future, and they will be made, the research community will have access to these, and who knows, Quanterix may even make a kit as well. And And we did come up with a sampling collection procedure for the isolation of PBMCs that the project team was able to utilize for a first-in-human exploratory study. And this accommodated all of the sampling site capabilities and preserved the integrity of the sample and also the phosphorylation state of the protein. So with that, just my acknowledgements. I'd just really like to highlight Rosemary Lawrence Henderson in my lab and Antonia Stepan in the Neuroscience Research Unit, and also Carlos Herman Cabrera from Quanterix. We're really glad to have him at the Andover Pfizer site. And also Samantha Hutton at Michael J. Fox Foundation has been extremely helpful throughout this process. So thank you for listening. So these are normal individual CSF samples that we purchased commercially. We were hoping to be able to measure endogenous levels in the CSF samples, and we were only to be able to measure 3 out of all of the samples that we detected. So the next step would be then to move to exosomes, probably.
There's no notion that tool is elevated in PD?
I believe in the literature it does say that it is elevated in PD, in CSF, but I don't know what tool was used to measure that, whether it was an ELISA or other type of platform. But we haven't looked at any PD CSF samples yet. Yeah.
We'll have questions for Alison as well. She'll be here for the reception.
I will, yes.
Well, thank you.
Okay, thank you.